Mutation, AQA biology. Key terms. Mutation, a change in the amount of the structure of DNA. Point mutation, change in one nucleotide at a particular point. Chromosomal mutations, gene deletion, insertion, and translocation, which we'll get into more into the PowerPoint. Whole chromosome mutation, an entire chromosome is lost or replaced in cell division. Tumor, a breakdown in cellular control mechanisms that put a brake on cell division. Cells rapidly divide and can occur anywhere, and they are made up of abnormal cells. Benign tumors, they grow large but don't destroy surrounding tissues. Malignant tumors destroy surrounding tissues and break away and invade neighboring cells, forming secondary tumors. Factors of mutation. You have gamma rays, X-rays, free radicals, incest, drugs, alpha and beta glucose, chance, and the sun rays. So this is just like a little diagram showing what a normal gene would look like and then what a mutated gene would look like. So it can either form the same protein or no protein at all. Mutation. Is it inherited? Well, in diploid cells, which are body cells, no. However, in haploid cells, which are gametes, yes. Causes, like in the previous slide, I talked about how it's random, radiation, chemicals which are mutagenic, and carcinogenic substances. So what are the different types? You have substitution, addition, deletion. Yeah, and there's more, but that will come up in A2, which you'll come up in another video. But this is just the first three that you need to know. So you have substitution, where a whole section of DNA, let's say this area, is replaced. Instead of GTA, we're gonna put in, I don't know, C. Okay, maybe this isn't the right place to draw this. Let's do it above. Okay. Instead of GTA, let's say we're gonna put, I don't know, ATG instead. That would be substitution. So we've changed the original sequence of G T A into a different sequence, which will code for a different amino acid, which will change the whole sequence. However, due to the code being degenerate, you don't know, you actually might form the same protein, depending on that. Then you have insertion, which is also known as addition. So let's say this is the sequence, and we actually insert, let's say, I don't know, a G, guanine. If we insert a guanine on there, it's going to cause a frame shift. And remember everything's read in triplet code. So it's going to cause everything after this point to be moved down by 1, which will change everything. So instead of coding for lysine, it might cause code for aline and completely change everything in the sequence. So a completely different amino acid will be coded for. And the one amino acid at the end will have one one codon that used to have 3 will now have 2 because 1 is already used up in the previous strand. So one at the end, there are going to be 2 non-functioning bases because they're not a triplet, so they won't be read. And deletion does the same thing. So we get rid of this diagram here shows that we get rid of a G, which will also cause a frame shift and have the same results as previously described. And the effects, they can be advantageous, like, you know, in like the Spider-Man films where you get bit by a radioactive spider and then you get superpowers. That would be advantageous because it puts you in a better position than the rest of the population. You can have no change because the code is degenerate, so I could just code for the same structure and have the same protein, or it could be fatal and actually kill you. Now, I hope you liked this video and I hope it was useful. And the next video will be about meiosis and genetic variation. So if you like this video, then please like and subscribe, and I'll see you on the next one.